Alright, you're welcome back. We are going to be considering questions 31 down, and if you missed the previous video, please go back and watch that video. The first part of it that covers from question 1 to question 30, because questions are repeated, and any one of the questions you miss, you might be missing the exam question. Alright, so question number 31. Dash is an organism in the phylum Coelentrata. So which organism is in the phylum Coelentrata? The answer is D, jellyfish. Jellyfish is under the phylum Coelentrata. Question 32. Dash example of an osteotides is dash. Osteotides, they are talking about bony fishes. Bony fishes. So looking at Pisces, that is the fishes, we have the osteotides, that is the bony fishes. We have agnata. Agnata is the jawless fishes, and we have the conjunctices, which are the cartilaginous fishes. So they are asking which one is an example of bony fish. That is what the question is talking about here. Which one is the example of bony fish? The answer is salmon. Salmon is a bony fish. Alright, now next question 33. Nematodes are dash organisms. Nematodes are dash organisms. So what are they? They are pseudo-coelomate organisms. They have false coelom. Anytime you hear pseudo means false. So they have false coelom. So that's why they say they are pseudo-coelomate. This is not coelomate, it's coelomate. Pseudo-coelomate. Alright. Now question 34. In the phylum Coelentrata, reproduction in Medusa is dash. Reproduction in Medusa is dash. Is sexual. So most of the organisms that fall under Coelentrata reproduce sexually. We'll move on to question number 35. Question 35. Adults of dash have radial symmetry while their larvae are bilaterally symmetrical. Alright, so the answer to that question 35 is actually A, Echinodermata. In Echinodermata, their adults, their body symmetry is radial, while their larvae have bilateral symmetry. So Echinodermata is the answer to that question. Question number 36. Crabs belong to the class dash. Crabs belong to the class dash. So the answer to that question is Arthropoda. They are arthropods. Crabs are arthropods. So under arthropods you see crustaceans and others. It's under arthropods you see crustaceans, you see insecta, and all those stuffs. So they belong here. Geese and sea goods belong to the class dash. That's question 37. The answer is aves. Aves, you know these things are birds, right? You know geese now and all those things, they are birds. So and birds are classified as aves. Question number 38. Question 38. Amphibians are represented by dash. So which of these are amphibians? I would advise anytime you don't know the options, especially when there are OBJ questions like this, use odd one out method. Check, is this one an amphibian? Is this one not an amphibian? So let's start from the known. Shark. Is shark an amphibian? No. You already know that shark is aquatic, right? Shark is a fish. So shark here is not the answer. Shark is not the answer because shark is classified under Pisces. Lizards have scales. So lizards are classified under reptiles. Snakes are classified under reptiles. Obviously you are left with one. You don't know newts. Maybe you don't know salamander. But this is obviously the only option remaining. So this is our correct answer. So you see how using odd one out method works. Using odd one out method works a lot. I use that a whole lot. So you remove those ones that seem very wrong, obviously wrong, then you'll be left with something that is the correct answer. Okay, that is why it's difficult to really fail OBJ exam if you use this trick. Now, arthropods, next question 39. Arthropods have dash. Arthropods have dash in question 39. A says respiratory organs, jointed appendages, exoskeleton, all of the above. The answer is obviously, okay, before I even tell you the answer, arthropods have respiratory organs. Ask yourself, if they don't have respiratory organs, how do they respire? Do they have jointed appendages? Maybe you don't know. But exoskeleton, you've heard that cockroach and other some insects, they have exoskeleton. So if these two, you've already known these two, then obviously this one should be there because there's no option that says A and C. So the answer is actually all of the above. So arthropods have jointed appendages, they have respiratory organs, and they have exoskeleton. Alright, next question, question 40. Please take these things very serious, very serious. Question number 40. In the history of classification, that's question 40 now. Question 40 now. In the history of classification, Dash proposed the idea of spatial creation, which lasted for nearly 2,000 years. Nearly 2,000 years. The answer is Carolus Linnaeus, the person that brought about binomial nomenclature. Next question. Albinism is an example of what factor of adaptation? What factor of adaptation? Albinism. The answer is actually mutation. Albinism is actually a mutation in one of the genes involved in thyroxine metabolism, particularly thyroxinase, the enzyme thyroxinase. Now, this mutation, you know, melanin is the skin pigment, right? What gives the skin coloration? Melanin. And that melanin is gotten from the metabolism of thyroxine, an amino acid. So if an enzyme, thyroxinase, involved in the metabolism of thyroxine is not there or is defective, melanin cannot be produced, and that is a typical case of albinism. So that's albinism. It's caused by mutation. The next question, question 42. Question number 42. Plants rooted in the bottom with leaves above the water line are called dash. You know, there are some plants that their roots are in the bottom of water, but their leaves are outside on the water line. So what do you call them? They are called elodates. They are called elodates. Take note of that, please. Question 43. One of these is not a feature of terrestrial habitat. One of these is not a feature of terrestrial habitat. It's not a feature of terrestrial habitat. You already know that terrestrial habitat, they're looking at habitat on land. High availability of nutrients. Is nutrients present? Desiccation. Desiccation. What is desiccation? What is desiccation? You ask yourself. Desiccation, loss of water, drying out, and abundance of oxygen. Is there oxygen? Is there light? Yes, there is oxygen. You mark that one. That one is not the wrong answer because the question says which is not. Abundance of light. Yes, so there is light. What do you think? There's light now. You are left with two options now. Now, how high availability of nutrients? Desiccation. Desiccation, drying out. So what are we choosing now? The answer is actually desiccation. It's not a feature of terrestrial habitats. Alright, so move on. Question 44. All but one of these are ecological conditions. All but one. When they say all but one, meaning they're asking you which of them is not. Which of them is not? So all but one of these are ecological conditions under which xerophytes strive. Now, xerophytes, zero means dry. Xerophytes are plants that live on dry soil, dry land, dry habitats like deserts. Those are xerophytes. So which of them, all but these are ecological conditions under, so which of them is not the ecological condition which xerophytes thrive? A says A says sorry, trying to take off this. All right, so that's done. So A says low soil, low soil moisture. B says strong wind. C says low temperature during the day. D, high sunlight. If you can see, two options here are contradicting each other. I don't know if you are seeing it. Two options here are clearly contradicting each other. And one way of knowing an answer is when two options contradict each other. If you look, C and D are contradicting. C says low temperature during the day. D says high sunlight. How can high sunlight, when sunlight is high, result to low temperature during the day? And we've already seen that xerophytes are dry plants that live in dry soil. So if the place is dry, then there cannot be low temperature during the day. So the answer is low temperature during the day is not. Remember the question says all but one, meaning which of them is not a condition for xerophytes. So it's low temperature during the day. During the day, there is always very hot sun, high temperature. Question number 45. Please click the subscribe button if you've not. That is the only thing you owe me. Please click on that subscription button. It is not MTN subscription. Nobody will charge you. You are not paying for it. You are not losing anything. So just help me click on that subscribe button. Thank you very much. As we move on, I wish you very good success. You come out of the hall smiling. Question number 45. Taiga is named after the biome in dash. So this one is a little common, so you should know. So, but what do you think? Russia. Russia is the answer. Russia is the answer. Question number 46. Derived savanna is likely not to be found in one of these states in Nigeria. Derived savanna is likely not to be found in one of these states in Nigeria. The answer is Benue. Who can tell me why? Question 46. Please drop your reason in the comment section why you said Benue. Drop your reasons in the comment section, but the answer is that's the answer. Question 47. The highest point in Nigeria is found in which state? The highest point in Nigeria is found in which state? It's found in Taraba State. It's found in Taraba State. Now, do you know what the highest point is talking about, Seth? You know we have land slopes and topography of the soil. The highest point is found in Taraba State. Ask those on Taraba State. If you are from Taraba State, let me know in the comment section. 48. Abuja is in what vegetational zone of Nigeria? Anytime you want to know vegetational zone, just ask yourself, that place they are mentioning, how is it climate? Remember, it is the climate that will determine the type of vegetation that is existing there. So is the place always dry? Is it always dry? Is it a rainforest? Is it always raining? If the place is raining, then it should be forest. Oh. If it's always dry, it may be savanna. Oh. It might be desert. Oh. You see now, with that you can help yourself. So, but the answer to this question, Abuja is in what vegetational zone of Nigeria? It's in the Guinea Savannah. Guinea Savannah. So we have three types of savanna here: Guinea Savannah, Sudan Savannah, and Sahel Savannah. Alright, so we move on. 49. Question number 49. Microscopic characteristics in bacteria include the following except dash. Microscopic, that is the underlying word there. Microscopic characteristics in bacteria include the following except dash. So it includes the following except what? In question number 49, the answer to that question is presence of flagella. Have you seen bacteria with flagella before? Flagella is used for movement in some organisms like euglena. Alright, but not in bacteria. Okay, so moving on. Question number 50. Question number 50 here. Question number 50. It says colonial characteristics of bacteria include all of the following except dash. Colonial characteristics of bacteria include all of the following except dash. The answer is depth of colony. We look at the shape of colony, we look at pigmentation of colony, edge or margin of colony, and depth of colony. Alright, so depth of colony is the answer to that question. Question number 51. The gram stain technique is an example of a dash. The gram stain technique is an example of a dash. It's an example of a differential staining technique. It's an example of differential staining technique. Alright, question number 52. Bacteria strains possessing capsules are usually dash. Those ones possessing capsules, they are very dangerous because now they are resistant to most of these antibiotics because they have capsules, something that is covering them, odeshi. So even when you take antibiotics will just be playing with them because they are resistant to it. So most bacteria strains possessing capsules are usually pathogenic. They cause diseases. Question number what now? Question number 53. Question number 53. If you are getting something, please click on the like button. Share this video to your class group so that others can also benefit. Question number 53. Sugar fermentation tests are an example of dash. Sugar fermentation tests are an example of dash. The answer is a biochemical test. Fermentation is conversion of sugar to alcohol. Have you ever heard that if you take palm wine when it is tapped, so if they tap palm wine now and you drink it, they say it's not alcoholic. It's not alcoholic actually because it's sugar. But if you allow it for maybe a day, it will ferment. Yeast will ferment it, will convert the sugar in it to alcohol, and you drink it, you start to be talking on that thing. Alright, so question number 54. Acid and gas production by bacteria in a sugar fermentation test is indicated by dash. So acid and gas production is indicated by what? A says change in color of the medium, B says effervescence, C a clear space in the inverted Durham tube. The answer is actually A and C. So the presence of acid and gas can be shown with a change in color of the medium, and a clear space in the inverted Durham tube. Durham tube is used for this test. Question number 55. Are we making progress? Yes. The basis of bacterial serological test is dash. Serological test, you're looking at antigen-antibody reactions. So unknown bacteria and anticellular reactions, protein and protein interaction, body serum antibody reaction, and none of the above. So let me explain a little here. Bacterial serological tests. Serological test, you want to test for antigen-antibody reactions using body fluids. So if you want to test for the presence of bacteria in body fluid, what is the principle behind that? That's what the question is saying. It's very crucial to understand the question. So it says unknown bacteria and anticellular reaction. Anticellular, the antibodies reaction. So unknown bacteria and antibody reaction, protein to and protein interaction, blood serum antibody reaction, and none of the above. The most correct answer here is unknown bacteria and anticellular reaction. You don't know the bacteria, so you study the reaction with anticellular and added antibody. We move on to the next question. Question 56. The technique where The technique whereby bacterial virus are used to identify bacteria is called dash. Bacterial virus used to identify bacteria. So what will you call that? What will you call that technique? It's actually phage typing. You've heard of bacteriophage, right? Bacteriophage are virus infecting bacteria. So they are called bacterial virus, bacteriophages. So you use phage typing to identify bacteria. Alright. The next question, question number 57. Which of the following organisms below is similar to organisms A, which is a G plus C ratio of 80%, which is a G plus C ratio of 80%. Now look at this question carefully. Say which of the following organisms below is similar to organisms A. And organism A, we've seen it which is as a G plus C ratio of 80%. So which is similar to that? The G plus C ratio here, you are looking at organisms that have high GC content, that shows high GC content. So if we have 80% here, 80% here of it, then just look at the one that is closest to it, that is similar to it, close to it, that is close to it. Example of those that can be close to this, you can look at, if you look at Mycobacterium tuberculosis, that one have around 65 to 68%. 65 to 68%. If you look at Corynebacterium, that one have 50 to 60%. 50 to 60%. But as it stands now, the closest to this is this. The closest to this is this. So that's the answer there. That's the answer we'll go with there. The next question. Next question, question 58. When restriction endonuclease enzyme out DNA, I think there's a mistake here, of unknown and known bacterial DNA into fragments and their lengths are compared with one another and form the basis of dash. Form the basis of dash. So if you look at this, restriction endonuclease, though there is a mistake in the question, but let's try to figure out what they are trying to say. Restriction enzymes are enzymes used to cut restriction sites so that new genes can be incorporated during biotechnology, genetic engineering. So, and if you look at polymerase chain reaction is just used to make multiple copies of a particular DNA. DNA fingerprinting, restriction endonuclease typing. The answer here is actually restriction endonuclease typing. Next question, ribosomal RNA sequencing involves dash. Ribosomal RNA sequencing. So if you are sequencing the ribosomal RNA, it involves dash. Comparison of the ribosomal RNA sequences of organisms, comparison of the transfer RNA sequences of organisms, comparison of the RNA sequences of organisms. All of the above. So we go with all of the above. The technique of Southern blotting is based on dash. The technique of Southern blotting is based on dash. So the answer here is actually nucleic acid hybridization. Nucleic acid hybridization. Alright, so we move on. The next question, that's question 60. Question 60. Okay, we've just answered question 60. Now move on to the next video to watch from question 61 down, and please don't miss out because you might be missing the very exam question. So see you in the next video where we look at 61 till finish, and we have many things to cover together. So don't fail to go back to the next video. See you there. All right.